there is a fish that should not exist. It is small, barely longer than a human hand. Its body is pale, almost translucent, like something made of glass and shadow. It has no scales. It has no armor. It has no sharp teeth, no venom, no bioluminescent lure to attract prey in the dark. By every measure we use to judge survival in the deep ocean, this creature should have been crushed, dissolved, or starved into oblivion long before anyone ever found it. And yet, in the year 2014, a team of researchers lowered a camera system into the deepest ocean trench on Earth, the Mariana Trench, and what came back on that footage changed our understanding of where life can survive. The fish was drifting calmly through water, so pressurized that it would flatten a nuclear submarine. It was feeding. It was thriving. It was against every reasonable expectation, alive. The depth at which this creature was filmed is difficult to comprehend. Imagine standing at the edge of a cliff and looking straight down into darkness. Now imagine that cliff is nearly 8,000 meters tall. That is more than 26,000 feet of water pressing down from above. At that depth, the pressure reaches roughly 800 times what we experience at the surface. 800 atmospheres. The weight of a column of ocean water, nearly 5 miles deep, concentrated on every square inch of that small, fragile-looking body. The pressure alone would kill a human in less than a second. It would crush bone, collapse lungs, and force air out of every cavity in the body before the brain even registered what was happening. But this fish, this pale and unassuming creature with a body like a tadpole, does not merely survive that pressure. It has made that crushing darkness its home. The creature was later formally described and named Pseudoliparis swiri, though most people now know it simply as the Mariana snailfish. It belongs to a family of fish called Liparidae, the snailfish, a group so obscure that most marine biologists barely mention them in textbooks. Snailfish are found in oceans all over the world, from shallow tide pools to deep trenches. But no one expected to find one thriving at the absolute limit of vertebrate life. No one expected it to be the top predator in its environment, and no one expected it to look so fragile, so soft, so utterly unequipped for the most extreme habitat on the planet. The Mariana snailfish is not a monster from the abyss. It does not have massive jaws or nightmare teeth. It is quiet, it is small, and it has survived in a place where almost nothing else with a backbone can. What does it mean when the deepest living fish on Earth looks like it could fall apart in your hands? What does it tell us about the rules we thought governed survival? For decades, scientists assumed that the crushing pressures of the Hadal Zone, the deepest layer of the ocean below 6,000 meters, would act as a barrier to complex vertebrate life. The thinking was straightforward. Pressure distorts proteins, it disrupts cell membranes, it interferes with the basic chemistry that keeps a body functioning. At a certain depth, the logic went, bones would become too soft, enzymes would fail, and no fish could sustain its biology under such relentless force. That assumption held for a long time. It held until a small translucent fish appeared on a deep-sea camera and quietly proved it wrong. By the end of tonight, you're going to understand why that single discovery forced scientists to rethink the limits of vertebrate life on this planet, and why the answers they found only raised deeper, more troubling questions about what else might be hiding in the 95% of the ocean floor that humans have never seen. Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It is a simple action, but it helps this channel reach more curious minds like yours. The story of the Mariana snailfish is not just a story about one small fish in one deep trench. It is a story about the limits of human knowledge. It is about the assumptions we carry into the dark and the way those assumptions shatter when we finally look closely enough. It is about a world beneath our feet, beneath our ships, beneath our understanding, where the rules are different, where the physics bends, and where creatures exist that we have not yet imagined. The ocean covers more than 70% of this planet's surface, and we have explored less than 5% of its floor. We have better maps of the surface of Mars than we do of our own seabed, and in the thin sliver of that ocean that we have managed to observe, we found something that should not be there. Now, let's begin. To understand why the Mariana snailfish stunned the scientific community, we first need to understand the world it lives in. Not just the Mariana Trench, but the ocean itself. The scale of the ocean is something the human mind struggles to process because we are land creatures. We think in terms of roads and mountains and buildings. We measure distance horizontally across plains and deserts. But the ocean demands a different kind of thinking. It asks us to consider depth. It asks us to consider what happens when you go down instead of forward, when the light fades, when the temperature drops, and when the weight of water above you becomes something close to geological. The average depth of the world's oceans is roughly 3,688 meters. That is about 12,100 feet. To put that in perspective, the cruising altitude of a commercial airplane is roughly 10,000 meters. The average depth of the ocean is more than a third of that altitude. But average depth is a misleading number. Much of the ocean floor sits at moderate depths, on continental shelves and gentle slopes. The real extremes, the places that redefine what we think we know, lie in the trenches. And the Mariana Trench is the deepest of them all. The Mariana Trench stretches across the western Pacific Ocean, east of the Mariana Islands, in a crescent-shaped scar that runs for roughly 2,550 kilometers. That is about 1,580 miles. If you laid the trench across the continental United States, it would reach from the coast of California to somewhere past the Mississippi River. But the trench's length is not what makes it extraordinary. It is the depth. At its deepest point, a narrow slot known as Challenger Deep, the ocean floor plunges to approximately 10,935 meters below the surface. That is nearly 11 kilometers, roughly 35,876 feet. Nearly 7 miles of water stacked on top of itself, pressing down with a weight that is almost beyond calculation. Here is the first comparison that helps make this depth real. Mount Everest, the tallest mountain on land, rises to 8,849 meters above sea level. If you took Mount Everest and dropped it into Challenger Deep, its peak would still be submerged by more than 2,000 meters of water. Over 6,600 feet of ocean would still sit above the tallest point on the planet's surface. The mountain would vanish completely. It would not even come close to breaking the surface. That is the scale of depth we are discussing. A place where the tallest structure on Earth becomes a minor feature on the ocean floor. The second comparison involves pressure. At the surface of the ocean, we experience 1 atmosphere of pressure. That is the weight of the air above us, roughly 14.7 pounds per square inch. For every 10 meters you descend into the ocean, you add approximately 1 more atmosphere of pressure. At 100 meters down, the pressure is about 11 atmospheres. At 1,000 meters, it is roughly 101 atmospheres. At the bottom of Challenger Deep, nearly 11,000 meters beneath the waves, the pressure reaches approximately 1,086 atmospheres. That is more than 16,000 pounds pressing on every square inch. Imagine taking a column of steel and compressing it from all sides simultaneously. Imagine stacking more than 1,000 cars on top of a single dinner plate. That is the kind of force that exists at the bottom of the Mariana Trench, every moment, without pause, without relief. The third comparison involves light, or rather, the absence of it. Sunlight, the energy source that drives nearly all life on the surface, penetrates the ocean to a depth of roughly 200 meters under ideal conditions. This upper layer is called the epipelagic zone, also known as the sunlight zone. It is where photosynthesis occurs, where phytoplankton bloom, where the colors of coral reefs are visible to the human eye. Below 200 meters, the light begins to die. Between 200 and 1,000 meters lies the mesopelagic zone, the twilight zone here. Only faint traces of blue light filter down, growing weaker with every meter. By 1,000 meters, the light is gone, completely. Not dim, not faded, but absent. From 1,000 meters to 4,000 meters, in a region called the bathypelagic zone, the only light that exists is biological, bioluminescence. Tiny flashes produced by living organisms, sparks of cold chemical light in an otherwise total darkness. Below 4,000 meters lies the abyssopelagic zone, the abyssal zone, where the water temperature hovers just above freezing and the darkness is absolute. And below 6,000 meters, beyond the abyss, lies the Hadal Zone, named after Hades, the ancient Greek realm of the dead. This is where the Mariana snailfish lives, in a world without light, without warmth, and under pressure that would obliterate nearly any machine humans have ever built. Consider how quickly the familiar world disappears as you descend. At the surface, the ocean is warm, bright, and full of motion. Waves roll, sunlight glitters, fish dart in every direction. Within the first 100 meters, you pass through the richest layer of marine life on Earth. Schools of tuna, pods of dolphins, the slow drift of jellyfish. By 200 meters, the light dims noticeably. The water turns from blue to deep indigo. The temperature drops. By 500 meters, you are in near total twilight. The creatures here have enormous eyes, adapted to capture every last photon of fading light. Some produce their own light, flashing bioluminescent signals in the gloom. By 1,000 meters, the last trace of sunlight is gone. You are in permanent night. The water is cold, between 2 and 4 degrees Celsius. The pressure is over 100 atmospheres. By 2,000 meters, the world has become silent, dark, and crushingly heavy. By 4,000 meters, you have entered the abyss. The seafloor here is covered in fine sediment, a rain of organic particles that drifts down from the surface like snow in slow motion. Marine snow, they call it. The decomposing remains of everything that has lived and died above. By 6,000 meters, you cross into the Hadal Zone. This is the realm of the trenches. The word Hadal was coined by the Danish biologist Anton Brun in the 1950s. He led the famous Galathea expedition, which was among the first to systematically sample life from the deepest parts of the ocean. Brun recognized that the trenches represented a distinct ecological world, isolated from the rest of the deep sea by extreme depth and pressure. He chose them carefully. In Greek mythology, Hades was the underworld, a place where the living were not meant to go. The Hadal Zone earned that name. Below 6,000 meters, the pressure exceeds 600 atmospheres. The temperature sits near 1 to 2 degrees Celsius. There is no light, there is no photosynthesis. Food arrives only as a slow, unpredictable rain of detritus from the world above, sometimes supplemented by the carcass of a whale or a large fish that sinks to the bottom and becomes a feast for scavengers. Life here depends entirely on what falls from above or, in rare cases, on chemical energy released from the Earth's crust. The history of human exploration of these depths is remarkably short. For most of recorded history, humans assumed the deep ocean was lifeless. In the 1870s, the crew of HMS Challenger, a British research vessel, conducted the first systematic survey of the deep sea. Between 1872 and 1876, the Challenger expedition travelled nearly 70,000 nautical miles, taking soundings and samples from the ocean floor. When they lowered their sampling devices into the deepest parts of the Pacific, they made the first recorded sounding of what would later be named the Mariana Trench. But their instruments were crude. Their understanding of the deep sea was limited. They found life at depth, which shocked the scientific community of the time, but they could not begin to explore the full extent of what lived in those pressurized waters. It was not until 1960 that a human being first reached the bottom of Challenger Deep. On January the 23rd of that year, Swiss oceanographer Jacques Piccard and United States Navy Lieutenant Don Walsh descended in the bathyscaphe Trieste, a deep-sea submersible designed to withstand extreme pressure. The descent took nearly 5 hours. When they reached the bottom at a depth recorded as approximately 10,916 meters, they peered through a small porthole into a world no human had ever seen. Piccard reported seeing a flat fish on the seafloor, though later analyses cast doubt on whether it was truly a fish or a sea cucumber. Their time at the bottom was brief, roughly 20 minutes, and the sediment stirred up by their landing obscured much of their view. But the achievement was monumental. For the first time, humans had proof that the deepest point on Earth was reachable. The question of whether anything lived there, however, remained largely unanswered. More than 50 years passed before another human returned to Challenger Deep. In 2012, filmmaker and explorer James Cameron made a solo descent in the Deep-Sea Challenger, a specially designed submersible built to withstand the full pressure of the trench. Cameron reached a depth of approximately 10,908 meters and spent several hours filming and collecting samples. His footage revealed a barren-looking landscape of pale sediment with occasional signs of life, including small amphipods, shrimp-like creatures that scavenged on the bottom. The dive captured worldwide attention, but it also underscored a humbling truth. After more than a century of oceanographic research, our direct observations of the deepest ocean amounted to a few hours of footage and a handful of samples. Between those manned dives, robotic explorers and unmanned landers did much of the work. In the 1995, the Japanese remotely operated vehicle Kaiko reached the bottom of Challenger Deep. In 2009, the Nereus hybrid remotely operated vehicle, built by the Woods Hole Oceanographic Institution, also reached the trench floor. These robotic systems could stay longer and observe more patiently than any human crew. They sent back images of amphipods, sea cucumbers, foraminifera, and strange microbial mats. But fish, true vertebrates with spines, were conspicuously absent from the deepest recordings. The assumption grew that vertebrate life simply could not exist at such depths. The pressure was too great, the food was too scarce, the chemistry was too hostile. That assumption held until a team led by researchers from the University of Washington and the University of Aberdeen decided to look more carefully. Using a free-falling lander, a device that sinks to the ocean floor under its own weight and records whatever passes in front of its cameras, they began a series of expeditions to the Mariana Trench. What they found there would overturn decades of accepted wisdom about the limits of life. They found a small, pale, translucent fish swimming calmly through water that should have destroyed it. They found the Mariana snailfish, and the questions that discovery raised were far more unsettling than the answers scientists were prepared to give. But what scientists discovered next was worse than anyone expected. The first time the Mariana snailfish appeared on camera, it did not announce itself. There was no dramatic entrance, no eerie glow cutting through the blackness. The footage shows a small, pale shape drifting into the frame with an almost lazy ease, hovering over the sediment like a ghost made of jelly. The camera was mounted on a Hadal lander, a device designed by marine biologist Alan Jameson and his team. The lander had been dropped into the trench as part of a broader survey of Hadal zoof life, equipped with Baton cameras to attract and document whatever creatures lived in that crushing darkness. At first, the researchers were focused on amphipods, the tiny crustaceans that were already known to inhabit the deepest trenches. Amphipods had been collected from Challenger Deep and other Hadal locations for decades. They were expected, they were documented. What was not expected was a fish. The footage was captured during a series of expeditions conducted between 2014 and 2017. The research was a collaboration between multiple institutions, with key figures including Alan Jameson of Newcastle University, later at the University of Western Australia, Thomas Linley of the same team, and Mackenzie Geringer, then a doctoral student at the University of Washington. The expeditions deployed Hadal landers at multiple sites within the Mariana Trench, at varying depths, to build a picture of the biological communities living at different levels of the trench. The cameras recorded footage for hours at a time, waiting in the dark for whatever might appear. The snailfish was filmed at depths between approximately 7,966 meters and 8,078 meters. That places it well within the Hadal zone, far below the point where most scientists believed fish could survive. Previous to this discovery, the record for the deepest living fish was held by another species, Abyssobrochula galathei, a cusk eel pulled from the Puerto Rico Trench at a depth of approximately 8,370 meters. But that specimen was dead when it was collected, dragged up in a trawl net in 1970. No one had ever observed it alive at depth. No one could confirm it truly lived at that depth, or had simply been caught in the net as it passed through shallower water on the way up. The Mariana snailfish, by contrast, was filmed alive, swimming freely, feeding on amphipods, and clearly at home in the Hadal zone. It was not a dead specimen pulled from a net. It was a living animal observed in its natural environment. Mackenzie Geringer, who would go on to lead the formal scientific description of the species, studied several specimens collected by the lander's traps. The fish were small, typically between 20 and 25 centimeters in length, roughly 8 to 10 inches. Their bodies were elongated, with a broad, flattened head and a tapering tail, giving them a tadpole-like silhouette. They had no scales. Their skin was thin and translucent, allowing the outlines of internal organs and muscle blocks to be visible through the body wall. Their skeletons were only partially mineralized, meaning their bones were not fully hardened like those of most fish. Instead, the bones were soft, cartilaginous in many places, more flexible than rigid. The skull, in particular, was so poorly ossified that it appeared almost fragile. Under a microscope, the bone structure looked incomplete, as if the process of building a skeleton had been started and then abandoned partway through. This skeletal structure was one of the first clues to how the snailfish survives at depth. Hard bones are a disadvantage under extreme pressure. A rigid structure, when compressed from all sides by hundreds of atmospheres, becomes a liability. It resists the pressure unevenly, creating stress points where fractures can occur. A softer skeleton, one that bends and flexes with the pressure rather than fighting against it, is far better suited to the Hadal environment. The Mariana snailfish's partially cartilaginous bones allow it to exist in a state of equilibrium with the surrounding water. The pressure pushes in from every direction, and the fish's body, lacking rigid points of resistance, simply conforms to that pressure. It does not fight the crushing weight of the ocean. It yields to it. But the skeleton is only part of the story. The deeper problem, the one that had convinced scientists for decades that fish could not live at such depths, involves biochemistry. Specifically, it involves proteins. Proteins are the molecular machinery of life. They catalyze reactions, transport molecules, provide structural support, and carry out virtually every function a living cell requires. For a protein to work correctly, it must be folded into a precise three-dimensional shape. That shape is maintained by a delicate balance of chemical forces, hydrogen bonds, electrostatic interactions, and hydrophobic effects. Under extreme pressure, those forces shift. The water molecules surrounding the protein are compressed, and they push against the protein's structure in ways that can distort or unfold it. When a protein unfolds, it stops working. It becomes a tangled, useless strand of amino acids. At the pressures found in the Hadal zone, the expectation was that most fish proteins would denature, that they would lose their shape and their function, rendering the organism unable to carry out basic biological processes. The solution turns out to be a molecule called trimethylamine N-oxide, often abbreviated as TMAO. This is a small organic compound found in the tissues of many marine fish, though typically in modest concentrations. TMAO has a remarkable property. It stabilizes proteins under pressure. It acts as a kind of chemical counterweight, pushing against the destabilizing effects of high pressure and keeping proteins folded in their correct shapes. The deeper a fish lives, the more TMAO it needs. Researchers had observed this pattern in fish at moderate depths. But the question was whether any fish could produce enough TMAO to survive at Hadal depths. When Geringer and her colleagues analyzed the tissues of the Mariana snailfish, they found extraordinarily high concentrations of TMAO, far higher than those found in any shallow water fish. The snailfish's cells were saturated with this stabilizing compound, essentially bathing every protein in a protective chemical shield, and it gets worse, or rather, more perplexing. The high concentrations of TMAO that protect the snailfish at depth may also define the boundary of where fish can live. TMAO is an osmolyte, meaning it affects the balance of water inside and outside cells. At very high concentrations, TMAO begins to alter the osmotic properties of the fish's tissues, making them more similar to seawater in terms of solute concentration. There is a theoretical limit to how much TMAO a fish's cells can tolerate before the osmotic balance becomes unmanageable. Researchers have calculated that this limit corresponds to a depth of approximately 8,200 meters, give or take a few hundred meters. Beyond that depth, the concentration of TMAO required to stabilize proteins would cause the fish's cells to lose the ability to regulate water flow across their membranes. The fish would, in essence, become chemically indistinguishable from the seawater around it. Its cells would lose their integrity. Life, at least vertebrate life as we understand it, would become impossible. This calculation is both elegant and deeply troubling. It means that the Mariana snailfish lives at or very near the absolute theoretical maximum depth for any fish. It is not just a deep-sea species. It is a species that exists at the edge of biological possibility. If we define the boundary of vertebrate life as the depth below which the biochemistry of a fish can no longer function, then the Mariana snailfish is pressed right against that wall. It is not merely adapted to extreme depth. It is adapted to the most extreme depth that its fundamental biology will permit. That is not an accident. That is something worth pausing over. The species was formally described and named in 2018 in a paper published in the journal Zootaxa. The name Pseudoliparis swire honors Herbert Swire, a sub-lieutenant aboard HMS Challenger during its famous expedition in the 1870s. It was Swire who recorded some of the deepest soundings of the Mariana Trench during that voyage, helping to establish the first measurements of a place that would not be visited by humans for nearly another century. The naming was a fitting tribute, connecting the modern discovery of the deepest living fish to the earliest attempts to plumb the ocean's greatest depths. But the discovery of the Mariana snailfish did more than set a record. It challenged a fundamental framework that scientists had used for decades to predict where life could and could not exist. The framework was based on pressure tolerance, protein stability, and metabolic constraints. It predicted a clear boundary, a line in the ocean below which no vertebrate should be found. The snailfish was found at that line, not comfortably above it, not cautiously distant from it, right at it. And the question that raised was immediate and unsettling. If life has pushed itself to the very edge of what is biochemically possible, what else might be happening near those edges that we have not yet seen? Consider the broader context of the discovery. The Mariana Trench is not the only Haddle environment on Earth. There are at least 33 known ocean trenches that reach below 6,000 meters. The Tonga Trench in the South Pacific, the Kermadec Trench near New Zealand, the Japan Trench, the Kuril-Kamchatka Trench, the Philippine Trench, the Peru-Chile Trench off South America, all of them plunge to Haddle depths. Each of these trenches is a unique environment with its own topography, its own sediment composition, its own patterns of organic input from above, and its own communities of organisms. Yet, as of the time of the snailfish's description, most of these trenches had been barely sampled. The Haddle Zone, which collectively represents roughly 2% of the ocean floor by area, is among the least explored environments on Earth. The number of scientific expeditions that have specifically targeted Haddle depths can be counted without reaching triple digits. The footage and samples that exist from those depths would fit into a modest library. Against the vast, dark, unknowable volume of the Haddle trenches, our knowledge is a single candle in a cathedral of darkness. The Mariana snailfish reminded scientists of something they already knew but preferred not to dwell on. Our understanding of the deep ocean is built on fragments. We lower cameras and landers to specific points, record what appears, and extrapolate from those observations. But the gaps between our sampling points are enormous. The area we have observed directly is an infinitesimal fraction of the total seafloor, and the deeper we go, the larger those gaps become. It is entirely possible, even likely, that species exist in the Haddle Zone that have never been seen, never been sampled, never been imagined. The snailfish was one of those species until a few years ago. It was living in the Mariana Trench for an unknown span of time, feeding, reproducing, dying, and being replaced by new generations, all in complete darkness, all under crushing pressure, all without any human being having the slightest idea it was there. There is also the question of how the snailfish got there. Snailfish, as a family, are found throughout the world's oceans. Some species live in shallow water, some live at moderate depths. A few, like the Mariana snailfish, live at extreme depths. The family appears to have diversified into deep water multiple times, with different lineages independently colonizing deeper and deeper habitats. But the Haddle Zone is not a continuous environment. Each trench is isolated from the others by shallower water, much as mountain peaks are isolated by valleys. A snailfish living in the Mariana Trench cannot simply swim to the Tonga Trench. The abyssal plains between the trenches, while deep, are significantly shallower than the trenches themselves, and the species that live in one trench may be genetically distinct from those in another. The Hadal trenches are, in a sense, islands, deep, dark, pressurized islands at the bottom of the sea, and the species that inhabit them may be as isolated from each other as the finches on the Galapagos Islands that captivated naturalists centuries ago. This isolation has profound implications. It means that each trench may harbour its own unique community of organisms, its own version of the Mariana snailfish, adapted specifically to the local conditions of that trench. When researchers from the HADES project, a large-scale investigation of Hadal ecosystems funded by the National Science Foundation, began surveying multiple trenches in the mid-2010s, they found evidence supporting exactly this idea. Different trenches contained different species of snailfish, different assemblages of amphipods, and different microbial communities. The Hadal zone was not a single uniform environment. It was a collection of isolated worlds, each with its own unique biology, connected by nothing but depth and darkness. The formal description of Pseudoliparus suaire made headlines around the world. News outlets called it the world's deepest fish. Scientists called it a window into the limits of vertebrate adaptation. But beneath the headlines and the records lay a more fundamental question, one that the discovery illuminated but did not answer. The question was simple. If we had been wrong about where fish could live, what else were we wrong about? But here's what makes this truly terrifying. The answer to that question is that we do not know. We do not know what other organisms might live at extreme depths. We do not know what adaptations they might possess. We do not know whether the TMAO boundary is truly the absolute limit or merely the limit of our current understanding. And we do not know what might be waiting in the trenches we have never sampled, in the waters we have never lit, in the sediments we have never disturbed. The Mariana snailfish was not hiding. It was simply living in a place we had never thought to look carefully enough. And that fact, more than the fish itself, is what should give us pause. The Mariana snailfish did not emerge from a vacuum of knowledge. Its discovery was made possible by decades of deep-sea exploration, each chapter of which pushed the boundary of what we knew and exposed the vastness of what we did not. To appreciate what the snailfish means, we need to examine the broader landscape of deep ocean mysteries, the explorations that preceded it, the discoveries that paralleled it, and the attempts humans have made to understand an environment that seems to resist understanding at every turn. The first mystery worth examining is the one that lies at the very foundation of deep-sea biology, the existence of life at hydrothermal vents. In 1977, scientists aboard the deep-sea submersible Alvin, operated by the Woods Hole Oceanographic Institution, descended to a depth of approximately 2,500 meters along the Galapagos Rift in the eastern Pacific. They were not looking for life. They were investigating geological features, hot springs on the ocean floor where superheated water, enriched with minerals from the Earth's crust, poured into the frigid deep sea. What they found around those vents was a biological community unlike anything known to science. Giant tube worms, some over 2 meters long, with no mouth and no digestive system, their bodies packed with symbiotic bacteria that converted chemical energy from the vent fluids into organic matter, clams the size of dinner plates, dense clusters of shrimp, crabs, mussels, an entire ecosystem thriving in total darkness, sustained not by sunlight and photosynthesis, but by chemosynthesis, the harvesting of chemical energy from the Earth itself. This discovery shattered one of the most basic assumptions in biology, that all complex life on Earth ultimately depends on the sun. Here was an ecosystem that had no connection to sunlight whatsoever. The bacteria at the base of the food chain used hydrogen sulfide and other chemicals from the vent fluids as their energy source. They were, in a sense, feeding on the planet itself. The organisms that lived around them, the tube worms, clams, and mussels, either hosted these bacteria inside their bodies or grazed on bacterial mats growing on the rocks. The entire community was powered by geothermal energy, by the heat and chemistry of the Earth's interior leaking into the ocean through cracks in the seafloor. No sunlight required, no photosynthesis, no connection to the world above. But here is the problem with hydrothermal vent communities. They are ephemeral. Individual vents have lifespans measured in years to decades, sometimes centuries, before the geological plumbing shifts and the flow of hot, mineral-rich water stops. When a vent dies, the community around it dies too, unless its members can disperse to another active vent. This raises questions about how these communities persist over long timescales. How do larvae from one vent reach another, potentially hundreds of kilometers away, through the vast food-poor expanse of the deep sea? The answer is still incomplete. We know that many vent species produce free-swimming larvae that drift in deep ocean currents, but the mechanics of dispersal, the survival rates, the navigation cues, these remain poorly understood. The vent ecosystems we have studied are likely only a tiny fraction of those that exist. Scientists estimate that only a small percentage of active hydrothermal vents have been discovered. The rest are out there, somewhere along the 65,000 kilometers of mid-ocean ridges that encircle the planet, pumping chemicals into the dark and sustaining communities we have never documented. The relevance to the Mariana snailfish is this. The vent discovery proved that the deep ocean contains entire ecosystems we did not know existed, functioning on principles we did not think possible. It proved that our assumptions about where life can and cannot exist were dangerously limited. The snailfish extended that lesson. If the vents showed us that life does not need sunlight, the snailfish showed us that vertebrate life can tolerate pressures we thought were beyond its reach. Both discoveries share the same underlying message. The deep ocean has rules of its own, and we are still learning what they are. The second mystery takes us to a different kind of deep-sea environment. The brine pools, scattered across the floors of certain ocean basins, including the Gulf of Mexico and the Mediterranean Sea, are lakes. Actual lakes, with defined shorelines, sitting on the bottom of the ocean. These are not ordinary bodies of water. They are pools of brine, water so saturated with dissolved salt that it is dramatically denser than the surrounding seawater. The brine is so dense that it forms a distinct layer on the seafloor, pooling in depressions and basins much the way water pools on land. The interface between the brine and the overlying seawater is visible, a shimmering, undulating surface that looks disturbingly like the surface of a lake seen from shore. Divers and remotely operated vehicles have filmed this interface, and the footage is surreal. It looks like an underwater shoreline, complete with waves rippling across the surface of the brine. The chemistry of these brine pools is hostile to most life. The salt concentration can be 3 to 8 times that of normal seawater. Dissolved methane and hydrogen sulfide are often present in high concentrations. The oxygen levels are extremely low or absent entirely. For most organisms, entering a brine pool is fatal. Fish that swim into the brine are overcome almost immediately, their bodies preserved in the dense, anoxic fluid like specimens in a jar. And yet, around the edges of these pools, life thrives. Dense colonies of mussels line the shore of the brine, their shells packed together in thick beds. Tube worms emerge from the sediment nearby. Specialized bacteria form thick mats on the brine surface, feeding on the methane and sulfide that diffuse upward from the pool. These are chemosynthetic communities, similar in principle to those at hydrothermal vents, but sustained by different chemical fuels. The numbers are striking when you consider the scale. The brine pool known as the Orca Basin in the northern Gulf of Mexico measures roughly 25 kilometers by 13 kilometers and sits at a depth of about 2,200 meters. The brine within it is nearly 8 times saltier than normal seawater. Its temperature is slightly above that of the surrounding water, and its chemistry creates an environment so toxic that nothing survives within the pool itself. But around its perimeter, on the narrow strip where brine meets normal seawater, an entire ecosystem flourishes. This pattern of death at the center and life at the margins repeats across multiple brine pools worldwide. It is as if the deep sea has created its own poisonous lakes, and life has found a way to colonize their shores without ever venturing into their depths. The third mystery involves sound. In 1997, the United States National Oceanic and Atmospheric Administration, using a network of underwater microphones called hydrophones, recorded an unusual sound in the deep Pacific Ocean. The sound was extremely loud, one of the loudest ever recorded in the ocean, and it had characteristics that initially suggested a biological origin. It rose in frequency over roughly 1 minute and was detected by sensors more than 5,000 kilometers apart. The sound was given the name bloop. For years after its detection, the bloop was the subject of intense speculation. If it was biological, what kind of creature could produce a sound that loud? The volume was far beyond anything known from any whale or marine animal. The speculation ranged from giant unknown cephalopods to undiscovered species of whale to creatures from the deepest trenches. In 2012, NOAA scientists concluded that the bloop was most likely the sound of a large icequake, the cracking and calving of a massive iceberg in the Southern Ocean. The acoustic signature, they determined, was consistent with ice-related seismic events, not biological sources. The mystery, in a sense, was solved. But the bloop served an important purpose beyond its own explanation. It drew public attention to the fact that the ocean is full of sounds we cannot identify, sounds that propagate across thousands of kilometers through the deep sound channel, a layer of water at intermediate depth where sound waves are trapped and can travel enormous distances with little loss of energy. The deep sound channel, centered at approximately 1,000 meters depth in most ocean basins, acts as a natural waveguide, bending sound waves back toward the center of the channel and preventing them from dispersing. It is through this channel that the songs of blue whales travel across entire ocean basins, and it is through this channel that the sounds of earthquakes, volcanic eruptions, and ice events are transmitted over continental distances. But here's the problem. While the bloop was identified as an ice event, the NOAA hydrophone network regularly detects sounds from the deep ocean that remain unexplained. Some have been given names: the slowdown, the whistle, the upsweep, Julia. These sounds have various characteristics, some rising, some falling, some periodic, some singular. Some have been tentatively attributed to geological processes, underwater volcanoes, seafloor movements, or ice dynamics. Others remain genuinely unexplained. The ocean is an acoustic world, a world of sound, and our ability to listen to it systematically is only a few decades old. The hydrophone networks cover only a fraction of the global ocean. What sounds are being produced in the Haddle Trenches, in the brine pools, along the mid-ocean ridges that we have never heard? The Mariana snailfish, living in a world of absolute darkness, likely relies heavily on vibration and pressure changes to navigate and find food. What acoustic landscape does it inhabit? We do not know. We have never listened carefully enough. The fourth mystery takes us to the sea floor off the coast of Japan, near the island of Yonaguni. There, at a depth of roughly 25 meters, lies a massive underwater rock formation known as the Yonaguni Monument. It consists of flat terraces, sharp edges, right angles, and what appear to be steps carved into the stone. When divers first encountered the structure in 1986, some researchers proposed that it was a man-made monument, possibly the ruins of an ancient civilizations submerged by rising sea levels thousands of years ago. The structures are striking. Some of the flat surfaces extend for dozens of meters. Certain features resemble columns, channels, and what could be interpreted as a road or a wall. The appearance is so regular, so apparently intentional, that the hypothesis of human construction seemed almost irresistible. But the scientific consensus has largely settled on a natural explanation. The rock at Yonaguni is sandstone and mudstone, which tend to fracture along straight lines and flat plains. The geological process of erosion, combined with the natural tendency of these rock types to break in regular patterns, can produce formations that look startlingly artificial. Similar natural formations have been documented on land, where flat-bedded sandstone erodes into terraces and right angles without any human intervention. The Yonaguni Monument, most geologists now agree, is a product of natural erosion shaped by the ocean, not by human hands, yet the debate has never been fully closed. A small number of researchers continue to argue for an artificial origin, pointing to features they believe cannot be explained by erosion alone. The site has never been systematically excavated or studied with the rigor that would be applied to a terrestrial archaeological site. The ocean, once again, makes thorough investigation difficult, costly, and slow. The relevance of Yonaguni to the broader story is not about whether it is man-made or natural. It is about the difficulty of interpreting what we see in the ocean. On land, we can walk up to a rock formation, chip away at it, analyze its layers, date its surfaces, and reconstruct its history with reasonable confidence. In the ocean, even at a modest depth of 25 meters, the investigation becomes exponentially harder. Currents erode evidence, marine organisms colonize surfaces, visibility is limited, equipment is expensive and time-sensitive. If it is this difficult to determine the origin of a structure in shallow water, imagine the challenge of understanding biological and geological processes at 8,000 meters, where no human can go, where cameras operate in total darkness, and where the only evidence is whatever happens to drift in front of a lens during the few hours a lander sits on the bottom. The fifth and perhaps most unsettling mystery involves the creatures themselves, the organisms that live in the deep sea and seem to defy the physical constraints we expect life to obey. The Mariana snailfish is one example, but it is far from the only one. In the Hadal trenches, researchers have found amphipods, small crustaceans at depths exceeding 10,000 meters. These are not vertebrates. They are invertebrates, and their biochemistry is subject to different constraints. But their abundance at extreme depth raises its own questions. Some of the amphipods collected from the Mariana Trench have been found with wood fragments and plant material in their guts, terrestrial organic matter that has somehow sunk to the deepest part of the ocean and been consumed by creatures living there. The connection between the surface world and the Hadal zone, it turns out, is not as severed as we might assume. Material from land, from rivers, from forests can travel through the ocean and reach the bottom of the deepest trenches. The Hadal zone is isolated, yes, but it is not entirely cut off. It receives inputs from the world above, inputs that we are only beginning to trace and quantify. Then there are the xenophyrophores, giant, single-celled organisms that can reach sizes of 20 centimeters or more. These protists, among the largest single cells on Earth, are found on the abyssal plains and in the Hadal trenches. They build intricate shells, called tests, from sediment particles cemented together with organic secretions. They are so large that they were initially mistaken for sponges or other multicellular organisms. Their biology is poorly understood. How does a single cell coordinate its activities over a body that large? How does it maintain structural integrity under the pressure of the deep sea? How does it feed, reproduce, and survive in an environment where food is scarce and the conditions are relentless? Each of these mysteries, hydrothermal vents, brine pools, unexplained sounds, enigmatic formations, and organisms that defy our expectations, converges on a single, uncomfortable conclusion. We have built our understanding of the ocean on assumptions that the ocean itself does not respect. We assumed that life needs sunlight. The vents proved otherwise. We assumed that toxic chemistry precludes complex ecosystems. The brine pools proved otherwise. We assumed that the deepest waters are silent. The hydrophones proved otherwise. We assumed that fish cannot survive below a certain depth. The Mariana snailfish proved otherwise. Each discovery peels back a layer of certainty and reveals a deeper layer of ignorance. Something is wrong with our assumptions, not in a catastrophic sense, not in the sense that everything we know is false, but in the sense that our knowledge is incomplete in ways that should make us profoundly humble. The deep ocean is not just unexplored, it is unexplored in a way that makes our exploration of it seem almost comically inadequate. We have sent 12 people to the surface of the moon. Fewer than a dozen human beings have ever reached the bottom of Challenger Deep. We have rovers crawling across the surface of Mars, sending back high-resolution photographs of alien deserts. We do not have equivalent coverage of our own ocean floor. The deepest places on our own planet remain more mysterious than the surface of other worlds. The Mariana snailfish sits at the center of this web of mysteries, not because it is the most spectacular creature in the ocean, but because it is the most quiet evidence of what we do not know. It was there all along. It was living, feeding, reproducing, and dying in the deepest trench on Earth, and we had no idea. It did not take a revolutionary technology to find it. It took a camera and a piece of bait. The technology to discover the snailfish existed for years before anyone thought to deploy it at the right depth, in the right place, with the right expectations. The limiting factor was not our equipment, it was our imagination. Here is a number that should stop every person reading this in their tracks. 95%. That is the proportion of the ocean floor that remains unexplored by human observation. 95%. Not of some distant planet, not of a theoretical dimension, of Earth, of the planet we live on, of the world we call home. More than 2/3 of the surface of this planet is covered by water, and of the seafloor beneath that water, we have directly observed, mapped, or sampled less than 5%. The rest is unknown, dark, unmapped, unseen. We have more detailed topographic maps of the surface of Mars than we do of the bottom of our own oceans. The rovers on Mars have sent back millions of high-resolution images. The deep ocean sends back almost nothing. Let that sink in. We live on a planet that is primarily ocean, and we have not looked at most of it. We have not photographed it. We have not sampled it. We have not sent cameras to its floor. The deep ocean, the largest habitat on Earth by volume, is effectively terra incognita, or more accurately, aqua incognita. Unknown water, unexplored, unlit, unwit. The reason for this ignorance is not a lack of curiosity. It is a matter of physics. The deep ocean is hostile to human exploration in ways that no other environment on Earth can match. Pressure is the most obvious barrier. At the bottom of Challenger Deep, the pressure exceeds 1,000 atmospheres. No unprotected human body can survive that. No standard submarine can operate at those depths. Even specialized deep-sea vehicles must be built with extraordinary care, using materials and designs that can withstand forces that would crush ordinary steel. The Trieste, the bathyscaphe that carried Pickard and Walsh to the bottom in 1960, used a float filled with gasoline, which is less compressible than water, to provide buoyancy and a heavy steel pressure sphere to protect its occupants. The entire vehicle weighed more than 50,000 kilograms. It was a marvel of engineering, and it could barely function at the bottom for 20 minutes. Beyond pressure, there is darkness. The deep ocean is the darkest natural environment on the planet. Below 1,000 meters, there is no sunlight, not a trace, not a faint glow, absolute darkness. Human eyes, even fully adapted to low light, cannot see anything in these conditions. Exploration requires artificial light, which must be powered by batteries or surface connections, both of which add complexity, weight, and cost. The darkness also means that traditional surveying methods, those based on visual observation, aerial photography, or satellite imagery, are useless in the deep ocean. Satellites can measure the surface of the ocean with remarkable precision, but they cannot see through kilometers of water to the bottom. Sonar, which uses sound waves to map the seafloor, can provide broad topographic data, but its resolution is far lower than optical imaging. A sonar map of the deep seafloor shows mountains and trenches in rough outline, but it cannot reveal the small-scale features that determine where organisms live, where sediment accumulates, and where the geology tells its stories. Temperature is another barrier. The deep ocean is cold. Below the thermocline, the layer of rapid temperature change that separates warm surface waters from the cold deep, the temperature drops to between 1 and 4 degrees Celsius. At Hadal depths, the water sits near 1 to 2 degrees, barely above freezing. This cold does not kill organisms outright. Many deep-sea species are well adapted to it, but it slows metabolism, chemical reactions, and biological processes. For human technology, cold water combined with high pressure creates challenges for seals, joints, electronics, and moving parts. Every component of a deep-sea vehicle must be designed to function in conditions that would immobilize most machines. And then there is distance. Not horizontal distance, but vertical. Getting to the bottom of the Mariana Trench requires descending nearly 11 kilometers straight down. The descent in the Trieste took nearly 5 hours. Cameron's descent in the deep-sea Challenger took roughly 2 hours and 36 minutes. Even the fastest free-falling landers take several hours to reach Hadal depths. Every expedition to the bottom of a trench is a commitment of time, energy, and resources. And when the vehicle or instrument arrives at the bottom, it can observe only its immediate surroundings. A camera on a Hadal lander illuminates a circle of seafloor perhaps 2 to 3 meters in diameter. Everything beyond that circle is invisible. To survey even a small area of the Hadal zone would require hundreds or thousands of lander deployments, each one costing time and money, each one recovering only a tiny fragment of data from an impossibly large domain. The result of all these barriers is a state of knowledge that is, by any honest assessment, shockingly thin. We know more about the moon's surface than about the floor of the Pacific Ocean. We know more about the topography of Venus, mapped by radar from orbit, than about the detailed terrain of most ocean trenches. The number of human beings who have walked on the moon is 12. The number who have reached the bottom of Challenger Deep, as of recent tallies, is fewer than that. More people have orbited the Earth in space stations than have descended to the deepest point of the planet they were orbiting. Let that sink in. The deepest point on our own world has been visited by fewer people than have travelled to an airless, radiation-bathed rock 250,000 miles away. This is not because the deep ocean is less interesting than the moon. It is because, in some fundamental ways, it is harder to reach. This is not science fiction. This is the current state of our knowledge. We live in an age of satellite imagery, global communication networks, and artificial intelligence, and we cannot tell you what lives on most of our own ocean floor. The Mariana snailfish existed in the Mariana Trench for an unknown span of time before anyone discovered it. How many other species are living in the deep ocean right now, at this moment, that no human has ever seen? The answer is certainly in the thousands, possibly in the tens of thousands. Every major deep-sea expedition in the past several decades has discovered new species. The rate of discovery shows no sign of slowing. If anything, as technology improves and more regions are sampled, the pace of new species descriptions is accelerating. We are not running out of things to find. We are finding out how much more there is to look for. The Hades project, which played a key role in the discovery and study of the Mariana snailfish, was one of the first large-scale efforts to systematically investigate Hadal Zone ecosystems, led by researchers including Timothy Shank of Woods Hole and Alan Jameson. The project deployed landers and trawls in multiple trenches across the Pacific, collecting samples and footage from depths below 6,000 meters. The results were revelatory. Each trench harbored distinct communities of organisms. Species found in one trench were often absent from another. The patterns of diversity were complex, influenced by depth, geography, food supply, and the physical shape of the trench itself. The Hades researchers found that the Hadal Zone was not a biological desert, as some had assumed, but a patchwork of isolated ecosystems, each with its own character and its own surprises. One of the most striking findings from the Hades project and related research was the abundance of life in the trenches. Hadal amphipods, in particular, were found in enormous numbers at bait stations. Thousands of small, shrimp-like creatures would swarm a baited lander within hours of its arrival on the trench floor. The amphipods were not just surviving at Hadal depths. They were abundant, active, and voracious. Some species had developed scavenging strategies that allowed them to detect and converge on food, falls from great distances, guided by chemical cues diffusing through the water. Their success at extreme depth was a reminder that life does not merely tolerate harsh conditions, it exploits them. Where there is energy, there is life. Where there is food, there are organisms to consume it. The deep sea follows this rule as reliably as any ecosystem on the surface. The Mariana snailfish, occupying the role of top vertebrate predator in this environment, feeds on those amphipods. It is a small fish eating small crustaceans, a quiet ecological interaction taking place in the most extreme habitat on the planet. There is no drama in it, no clash of titans, just a pale translucent fish gliding through the darkness, picking off amphipods that are themselves busy scavenging whatever has drifted down from the sunlit world above. The food web of the Hadal Zone is simple compared to those at the surface, but it is functional, self-sustaining, and, in its own way, complete. Primary production, in the form of chemosynthetic bacteria, supplements the organic rain from above. Amphipods and other scavengers convert that organic matter into biomass. The snailfish converts amphipods into vertebrate tissue. Decomposers break down what remains, recycling nutrients back into the system. It is a functioning ecosystem, in the deepest, darkest, most pressurized environment on the planet. The deeper truth, the one that the Mariana snailfish embodies, is not about one fish or one trench. It is about the fundamental inadequacy of our picture of the natural world. We draw lines on maps, we establish categories, we define zones, We say life can exist here but not here. Vertebrates can tolerate this depth, but not that depth. The ocean is explored enough for us to understand its general patterns, and then a small translucent fish appears on a camera at 8,000 meters and quietly reminds us that our lines are preliminary, our categories are provisional, and our understanding is a work in progress. The ocean does not care about our categories. It fills every niche, it pushes every boundary, it contains multitudes that we have not begun to count, let alone comprehend. The philosophical weight of this realization is considerable. We are a species that has mapped genomes, split atoms, and sent spacecraft beyond the edge of the solar system, and we do not know what lives on the floor of our own planet. We do not know the full extent of life beneath our own feet. The Mariana snailfish is proof that the unknown is not somewhere far away, in some distant galaxy or hypothetical dimension. The unknown is here. It is deep, it is dark, and it is wet. What does it mean for humanity that the deepest living fish on Earth was unknown to us until a few years ago? What does it mean that we had to lower a camera into the darkness, bait it with a piece of mackerel, and wait for hours just to catch a glimpse of a creature that had been living in the Mariana Trench long before we ever thought to look? The answer touches on something larger than marine biology. It touches on the nature of knowledge itself, and on humanity's peculiar relationship with the planet it inhabits. We are, as a species, remarkably good at looking outward. We have telescopes that can detect light from sources billions of light years away. We have probes that have left the solar system. We have sent robots to the surface of Mars, to the atmosphere of Venus, to the orbit of Saturn. We photograph distant nebulae with exquisite clarity. We listen for radio signals from other civilizations. We look up, we look out, we look away, and beneath us, covering more than 70% of our planet's surfaces, lies an abyss that we have barely glanced at. The imbalance is extraordinary. Our gaze is fixed on the heavens, while the ocean beneath our ships remains a mystery. This is not a criticism of space exploration. The impulse to look outward is a magnificent expression of human curiosity. But it is worth noting the asymmetry. We have invested orders of magnitude more money, more time, and more technological effort into exploring space than into exploring the deep ocean. The reasons are partly practical. Space exploration generates technologies and knowledge with broad applications, and partly psychological. Space is romantic. It is visible. We can see the moon from our backyards. We can point at Mars in the night sky. The deep ocean offers no such spectacle. It is invisible, hidden beneath a surface that looks the same from above, whether the water below is 10 meters deep or 10,000. The deep ocean does not inspire the same awe because we cannot see it. It does not fire the same imagination because we cannot easily picture it, and so we neglect it, not out of malice, but out of a failure of attention. The discovery of the Mariana snailfish is a corrective to that failure. It forces a moment of reckoning. Here is an animal, a vertebrate, a creature with a spine, a brain, a beating heart, living in a place we assumed was beyond the reach of its kind. And we found it not through some grand expedition costing billions of dollars, but through a relatively simple deployment of a baited camera on the ocean floor. The technology was available for years before the discovery was made. The capability existed. What was missing was the will and the vision to deploy it in the right place. There is a pause required here, a moment to consider the weight of that fact. The Mariana snailfish was not hidden by technology. It was hidden by assumption. The assumption that fish could not live at Hadal depths discouraged researchers from looking for fish at Hadal depths. The absence of evidence was taken as evidence of absence. And for decades, that circular logic held. No one looked, so no one found, so everyone assumed there was nothing to find. The snailfish broke that cycle, but how many other cycles remain unbroken? How many other assumptions are keeping us from seeing what is right in front of us, or rather, right below us? The implications extend beyond biology. The deep ocean is a reservoir of chemical compounds, genetic resources, and geological knowledge that we have barely begun to tap. Organisms living in extreme environments often produce unique biochemical compounds, enzymes that function at extreme pressures, proteins that resist denaturation, molecules that have no analog in surface life. These compounds may have applications in medicine, in industry, in biotechnology. The enzymes from deep-sea organisms have already found uses in molecular biology laboratories around the world. But the vast majority of Hadal Zone organisms have never been collected, never been cultured, and their biochemistry remains entirely unknown. What medicines might be derived from creatures we have not yet found? What industrial processes might be improved by enzymes we have not yet isolated? These are not fanciful questions. They are practical ones, grounded in the demonstrated reality that deep-sea organisms produce novel biochemistry. There is also the matter of climate. The deep ocean plays a critical role in the global carbon cycle, absorbing carbon dioxide from the atmosphere and sequestering it in deep water and sediments. The biological pump, the process by which organic matter produced at the surface sinks into the deep sea, transfers vast quantities of carbon from the surface to the abyss. The organisms of the deep ocean, from bacteria to amphipods to snailfish, are participants in this process. They consume, transform, and recycle the organic matter that falls from above. Understanding how the deep ocean food web functions, how efficiently it processes carbon, how it responds to changes in surface productivity, is essential for predicting how the ocean will respond to changing conditions in the atmosphere and climate. And yet, our knowledge of deep ocean ecosystems remains fragmentary. We are trying to understand a planetary scale process while missing most of the data. The Mariana snailfish, in its own small way, is a reminder of everything we are missing. It is one species in one trench. There are dozens of trenches, thousands of miles of mid-ocean ridges, millions of square kilometers of abyssal plain. And in all that darkness, in all that cold, under all that pressure, life is happening. Creatures are being born, are feeding, are reproducing, and are dying, all without any human witness. The deep ocean is the largest habitat on Earth, and it is living its life without us. We are not part of its story. We are at best occasional visitors who stay for a few hours and then retreat to the light. That should humble us, not in a way that diminishes human achievement, but in a way that clarifies human perspective. We are not the masters of this planet. We are inhabitants of its surface, clinging to a thin layer of rock and air while below us an entire world unfolds in silence and darkness. The Mariana snailfish does not know we exist. It does not need our approval or our attention. It has been surviving at 8,000 meters without us, and it will continue to do so whether we study it or not. We are the ones who need the snailfish. We need it as a reminder, as an anchor for our humility, as proof that the universe, even the small corner of it we call home, is far more complex and far more surprising than we tend to assume. In the end, the Mariana snailfish is a quiet revelation. It is not a monster. It is not a leviathan from the deep. It is a small, translucent fish, barely the length of a human forearm, living in the darkest and most pressurized habitat on the planet. It has no armor. It has no weapons. It has no bioluminescent display to advertise its presence. It is soft. It is pale. It drifts through the darkness like a thought that has not yet fully formed. And it is, by any measure we can apply, the deepest living fish on Earth. What the Mariana snailfish teaches is not a lesson about toughness or aggression or dominance. It is a lesson about adaptation, about the quiet ingenuity of life finding a way to exist where it should not be able to exist. It is a lesson about flexibility, about the advantage of softness over rigidity, about yielding to pressure rather than resisting it. The snailfish's partially cartilaginous skeleton, its tissues saturated with trimethylamine N-oxide, its slow metabolism, its simple diet of amphipods in the darkness, these are not dramatic adaptations. They are subtle ones. They are whispered solutions to screaming problems. And they work. They work at a depth where the pressure would crush a submarine, where the darkness is absolute, where the cold is relentless, and where the food arrives only as a slow, unreliable rain from the world above. The deep ocean does not reward spectacle. It rewards persistence. It rewards the ability to do more with less, to survive on scraps, to function in conditions that would destroy more conspicuous forms of life. The Mariana snailfish embodies that principle. It is not the strongest creature in the ocean. It is not the fastest, or the largest, or the most fearsome. But it is there. It is there at a depth where nothing with a backbone is supposed to be. And its presence, quiet as it is, carries the weight of a profound truth about the planet we inhabit. That truth is this: we do not know our own world. We have mapped its coastlines, named its mountains, charted its deserts, and catalogued its forests. We have built cities on its surface and sent signals through its atmosphere. But beneath the water, beneath the waves that we watch from shore, beneath the ships that carry our goods and our ambitions, there is a world that remains almost entirely unknown to us. The Mariana snailfish is one small window into that world, one small proof that the darkness below holds secrets we have not begun to uncover. The ocean does not reveal itself willingly. It demands patience. It demands humility. It demands that we set aside our assumptions and look with fresh eyes at a world that operates on principles we are still learning. The Mariana snailfish was there before we found it. It will be there after we stop looking. It does not exist for our benefit or our education. It exists because life, in all its forms, reaches for every possible niche, fills every available space, pushes against every boundary, and finds a way. And somewhere in the darkness below, in the trenches we have not explored, at depths we have not reached, in waters we have not lit, there are other secrets, other creatures, other adaptations, other truths, waiting, not for us, simply waiting, simply existing, simply alive. The deepest fish in the ocean is not a monster, it is a whisper. And if we are wise enough to listen, it tells us everything we need to know about how little we understand the world beneath our feet.